Chromosomal aberrations are variations in the structure of chromosomes. They usually affect many genes at a time and will play a major role in an individual's phenotype. There are several kinds of chromosomal aberrations, including deletions, duplications, etc. Let's take a look at a couple of examples. This first individual has a pair of chromosomes with gene sequence as listed here: GHI centromere JKLM and GHKJ centromere ILM. If we take those two chromosomes and stretch them out side by side, we can see that the middle region is reversed on the blue chromosome. This is called a pericentric inversion because it involves a centromere. What happens when the cell goes through meiosis? The chromosomes duplicate and create two sister chromatids. Then they pair up for the first meiotic division so that the genes are coupled accordingly. However, in this case, if the chromosomes were to just line up, the genes would not match each other. So in order to match the greatest number of genes together, an inversion loop is formed, and that's what we see here. The chromosome that contains the inversion loops around itself and fits in the dome created by the chromosome with a normal gene sequence. Let's look at a diagram to see what would happen if crossing over occurred between J and K. Here are paired chromosomes with the inversion loop. The crossing over will only affect the sister chromatids that are touching each other. To figure out what gametes will be created from this, we can use a highlighter and follow the chromatids from one end to the other. The chromatids highlighted in red and green are the ones that will not be affected by the crossover. The gametes they produce will be the same as the original chromosomes. The yellow and purple chromatids will be affected by the crossover. The gametes produced will be GHI centromere JKHG for yellow and MLKJ centromere ILM for purple. These last two gametes have genes that show up twice in the same chromosome, so they're not viable. The second individual has a pair of chromosomes with this gene sequence, GHKJ centromere ILM. Both chromosomes in this case have the same order of genes. When this individual goes through meiosis, the chromosomes will duplicate and create the sister chromatids. Then chromosomes are paired up for the first meiotic division like so. Here's what would happen with crossing over. It will only affect the sister chromatids in the center. If we highlight the chromatids again, we see the red and green chromatids are the ones that will not be affected by the crossover. The gametes they produce will be exactly the same as the original chromosomes. The yellow and purple chromatids will be affected by the crossover, producing gametes that will also have the same sequence as the original chromosomes. In this case, though, part will have come from the pink chromosome and part from the blue chromosome. We can see the red and green gametes have the original sequence, each from one of the parent chromosomes. The yellow and purple look like the original but are recombinant chromosomes. None of them have any duplicated genes, so they're all viable gametes. For that reason, individual 2 has a higher fitness than individual 1 because all of their gametes are viable.